Cell cycle is the mechanism by which all cells are duplicated and divided. It is consisted of 4 phases: G1, S, G2, and M. Chromosomes are duplicated during S phase, and then daughter chromatids are segregated during M phase for both daughter cells to receive the same number of chromatids. During mitosis, chromosomes line up at the equator of the cell. Correct biorientation happens as microtubules extending from opposite poles of the cell attach to kinetochores of sister chromatids and pull them apart. Attachment of microtubules to kinetochores might be faulty, and both sister kinetochores can be attached and pulled by the microtubules extending from the same pole. If such errors are not prevented by the cell, they can cause abnormalities and defects such as Down syndrome and cancer. Cells identify aberrant attachments as no tension is applied. RRB is a protein kinase that, by phosphorylating kinetochore components, will weaken and remove such attachments to be replaced with correct attachments and established biorientation. M1 and NDCAT complexes are crucial components of kinetochore in a kinetochore microtubule attachment, and it was known from previous in vitro studies that N-terminus of NDCAT and C-terminus of DAM1 are phosphorylated by aurora B for breaking up aberrant attachments. For a new attachment to form, kinetochores are first captured by the lateral surface of microtubules. Microtubules will then shrink until lateral attachment is transformed into an end-on attachment. To find out more about this correction process, we interviewed Professor Tomotanaka at the Center of Gene Regulation and Expression at University of Dundee to ask him a few questions about the findings of his lab on this topic.
So this kinase, aurora B kinase, is important to remove this wrong kinetochore microtubule attachment, but it was not known how then fresh attachment is still formed. So if aurora B is kind of weakening and disrupting wrong attachment, the new attachment must be also discouraged by this kinase. But the new attachment must happen efficiently, otherwise this turnover of kinetochore microtubule interaction wouldn't occur. And then this turnover is necessary to establish this correct orientation, which is called chromosome biorientation.
Tanaka Lab investigated how the new correct attachments can be formed despite the presence of aurora B kinase in vivo using Saccharomyces cerevisiae as their model organism. They investigated the importance of NDCAT N-terminus and DAM1 C-terminus domains in both lateral and end-on attachments by deleting those two domains from their protein complexes. Their observations suggested that after deletion of these two domains, the end-on attachments were detached and biorientation could not be reached. However, the lateral attachments still occurred in their absence. This proved that lateral attachments are not dependent on these two domains, but it was evident that they played a key role in the conversion of lateral to end-on attachments. They further analyzed the behavior of these attachments when NDCAT N-terminus and DAM1 C-terminus are mutated to become negatively charged as if they were phosphorylated by aurora B. They found out that they showed the same phenotype as the deletion of the domains. End-on attachments were disrupted and caused difficulty in reaching biorientation, while the kinetochore interaction with microtubule lateral surface remained unaffected.
So it was known that kinetochore can interact with the side of microtubule, and then it interacts with the end of microtubule. And then in this work, we found that this lateral attachment and then this end-on attachment are differentially regulated by this aurora B kinase. And then that's the kind of key how this turnover of kinetochore microtubule interaction still promoted by aurora B kinase. So that was an exciting finding.
Based on observations, it was concluded that as lateral and end-on attachments are differentially regulated, fresh lateral attachments are still formed and unaffected in presence of aurora B kinase to establish biorientation.